All right, so right now we're going to be looking at fermentation by yeast experiment in module 2. I didn't do the first 2 experiments on video because it's literally looking at slides and then also creating your cell, and everybody's cell is going to look different. So I'm only recording the ones that students have trouble with. So for this lab, the fermentation by yeast lab, the materials that you need is you're going to have 3 packets of sugar, 1 equal, 1 Splenda, 1 regular sugar packet. You're going to have a 1% glucose solution that came in your kit. Actually, all of this came from your kit. And then a 1% sucrose solution. That's not to be confused by this 0.5 one. This one is for something else. So you want to use the little bottle. You also need to have 5 beakers, 250 milliliter beakers. You can tell that by looking at the top number. You need to have the test tube rack and 5 glass test tubes. Be really careful with these. They have a habit of breaking. You need 5 of the small plastic test tubes. You need to have 4 pipettes, a graduated cylinder, a ruler. They say to write with permanent marker. I don't like to do that. I would use the dry erase marker, so that way you can always get the numbers and the letters off if you need to. And then it tells you that you're going to need warm tap water, measuring teaspoon, and a stopwatch. I always just use my phone, but if you have a timer that you want to use, that's fine. The warm tap water is just warm water out of your sink, and yeah, 1 measuring teaspoon. You'll have to provide that. Okay, we're going to get started in just a second. Okay, so the first thing that you want to do in any lab is I like to print out the directions. Now you can have your computer open doing directions, but that can be a little hazardous because if anything spills or goes wrong or erupts, you have an electronic that's, that's not really conducive to that electronic. So I like to have them printed. So I have my printed copy of instructions right here. And it also has a little checklist so that you can just go and check off as you go through. So the very first thing that you want to do is read through the directions multiple times, making sure that you understand each step that you have to do. And then I like to label as I go along. So whenever I'm reading through the instructions and it tells me, okay, you need to label these things, I label them before I start the lab. So in these instructions, it wants you to have a beaker labeled equal, 1 beaker labeled with Splenda, 1 beaker labeled sugar, and 1 beaker labeled yeast. And I just use the dry erase marker to do that. Then you're supposed to be labeling the test tubes. I labeled them at the bottom so that way you can see the numbers through the bottom of the test tube rack. So you can see that one says 2, that one says 3. And then they want you to label the plastic test tubes as well. So I'm just going to do that real quick. And the labels correspond with the solutions that you're going to be putting in there. So this is just to avoid any confusion that comes up, because you could be looking at it and saying, oh wait, what's number 4 again? And that'll be on your instructions. So this just kind of keeps everything organized. And like I said, I like to use the dry erase because it comes off, and since you're going to be using these test tubes and beakers in numerous experiments, just to avoid confusion, you can see it just comes right off, whereas the permanent marker doesn't do that necessarily. So there we go. Okay. All right, so we're going to go ahead and get started. So after you have your labeled, it asks you to empty the equal, the Splenda, and the sugar packets into the beakers. So here goes the equal. Splenda. Make sure you get all of it out because you're making solutions with this. So you want to have all of the materials in the beaker. Okay. And then I went ahead and got some warm tap water before I started this. And so they want you to fill the equal and the Splenda beakers to the 100 milliliter mark on your beaker. So you just want to take, and it, oh gosh, yep, see, accidents happen. You just want to take your warm tap water. It has to be warm or else the sugar won't dissolve. And we're making a solution, so we don't want sugar crystals floating around. So I'm just going to fill it to the 100. Same thing here. Just fill it to the 100. Okay. So now we're on step 5 of our instructions. And the sugar beaker, they actually want you to fill to the 200 milliliter mark. And that's just because the sugar amount is a little bit different than the other two. Oh, look, I did that pretty well. That was unintentional. Had exactly the amount of tap water I needed. Okay, and so then they want you to mix it by pipetting the solution up and down, and don't use the same pipette, because then if you do that, you're getting different sugars in the solutions, and that's going to mess up your experiment. So just take one pipette and you just squeeze and then squeeze it out. So squeeze it up, squeeze it out. Just a whole bunch of different times. Now the important thing is that you shouldn't see any sugar crystals. They should have dissolved in the hot water. Okay. So you can kind of mix it around too. All right, and we're just going to leave that pipette in there for right now, so that way we know not to cross-contaminate. Whoops. So here is the Splenda. We're just going to pipette it up and down a little bit. Now, if any of you have kids, this is kind of fun to do. My kids are out in the yard right now. They were threatened not to come in while I was filming. But yeah, if you have kids like this, you can get them involved in this if you want. This is kind of fun for them. Okay, so there's the Splenda. And then your sugar. Okay, now my sugar, I can still see some crystals, but that's okay. It's looking like it is still mixing okay. So my water might have cooled off a little bit before I got to the sugar. So you want to make sure your water is hot. Otherwise it's not going to dissolve like you want it to, but there. After a couple of pipetting, it worked pretty well. Okay, so your solutions are ready to go, and now each beaker has a 1% solution. So this is 1% equal, 1% Splenda, and 1% sugar. There's a calculation that we're not going to get into on how to make a 1% solution, but just know that that's what these are. All right, so we're on step 7, where it wants you to fill a small plastic test tube with tap water. I just put some tap water into the graduated cylinder, and so I'm going to pour it just real slowly into the plastic test tube. I know my hand's kind of in the way. And I'm just going to fill it all the way to the top. Now you don't have to overflow it. And what you will do is it wants you to take one of the large glass test tubes and turn it over. And then you're just going to push the small plastic test tube up inside the glass test tube all the way to the top. So the top of the plastic one should be as far up as you can push it. And then they want you to invert it. So basically you're just taking the glass test tube. Now you can see my finger is holding the plastic test tube inside the glass one, keeping it steady. And then you're just going to invert it. So you're just going to turn it upside down and you'll see a bubble went up to the top. That's what you want to see. And then you can take your finger out and now you have the fermentation tube. Now whenever you're ready for it to come back out, you just put your finger back down and then just flip it. And then you'll take it out in a sink because it'll splash everywhere obviously and get really messy. And we don't want to do that to the table right here. So you can just practice that a couple of times. Filling the tube, putting it inside, and then inverting it. But like I said, each time that you invert it, you should see this bubble that comes to the top. All right, so the next part of this experiment, they want you to fill your yeast beaker with 175 milliliters of warm tap water. You don't want it like scalding hot because that'll kill the yeast, so just warm enough for it to mix. I did that by getting 100 milliliters from my graduated cylinder and pouring it into the beaker, and then I found 75 milliliters and pouring it in there. So that's 175. You want to be precise with this whenever you do these labs. It's really important to be as close to the mark as you can be. Otherwise, it can mess things up. And so now they want you to take a teaspoon of yeast. So I have a teaspoon measure right here. And go ahead and pour it into that yeast beaker. And then take your last pipette, the one that hasn't been used, and just kind of mix it around. Okay, I like to stir at first. And then you can start pipetting it up and down until you get kind of that homogeneous mixture to where it's mixed in kind of like you did with the sugar. Now you don't want to leave this for too long because then you can have yeast settling to the bottom, and that's a no-no because you want to have yeast inside the tubes. So this is almost mixed. Like I said, sometimes you can just use the pipette like a spoon and just kind of, while you're pipetting up and down, you can just stir it. Okay, so hopefully you've already labeled your tubes because taking time right now to label them is not a good idea. They should already be labeled. And it says that you're gonna measure and pour 15 milliliters of each of these solutions into the plastic test tube. So the glucose solution is going to come from the glucose bottle, and the sucrose solution is going to come from the sucrose bottle. So we need 15 milliliters, and the way that you should do that is I would not use this large graduated cylinder. Instead, I would use the smaller one that came with your lab kit that of course now I can't find. Oh, here it is. Because it's such a small amount, you don't want to, you can overshoot it by putting it in the big one, but it's up to you. So, well, I guess, yeah, that is probably too small. You're going to end up spilling it. So yeah, go ahead and use the bigger container. So we're going to go with the glucose solution first. And if you want to speed it up through this part, that's fine. You want to hold the graduated cylinder at eye level, and you want to get to what's called the meniscus. So 15 is at that halfway point between 10 and 20 on your graduated cylinder. There we go. Okay. So you want it right at that middle. Now I poured a little too much, but not too bad. So it's at least 16. And so I'm going to put it into the small test tube. Now here's where they should have given you more than one test tube rack, because once you pour them into the test tube, what do you do with them without them spilling out? So what I did was I got an extra beaker, and I'm just gonna set them in there, and they're already labeled, so you shouldn't confuse them. Okay, and so, right, I used my graduated cylinder. I have to rinse this out now because you don't want to contaminate it with the other solutions. So whenever I come back to the video, my test tubes are already gonna be poured. Okay, so now we're at where we have all of our plastic test tubes filled with each solution, making sure that you rinsed the graduated cylinder after every time that you put a solution into the plastic test tube so that you didn't cross-contaminate it. So now there's no Splenda and equal mixed together. They're all in the same, they're all in their individual test tubes and not contaminated. So the next part of the experiment is on number 13, and it wants you to fill each space in the plastic test tubes with the yeast solution. So here's your yeast solution. Just mix it a little bit more to make sure that there's nothing that's at the bottom of the beaker. And you want to do this part fairly quickly. So have your glass test tube ready to go, and you're going to take your first test tube, the one that you labeled with a 1, and they're kind of mixed up in here a little bit. There we go. Okay. And you're just going to fill, so see how there's a little bit of space at the top? You're going to take your pipette, and you're going to put the pipette at the bottom of the beaker, and then just pull some yeast up. And you're just going to fill the rest of the tube almost to the top with this yeast solution. Take your glass tube. Remember, push the plastic tube all the way to the top, and then you're going to invert it. Okay? And so what it wants you to do is measure the initial air space. So the initial airspace is the air that's at the bottom of the tube to the top. And so you should measure this in centimeters. So when I measure it, it's about 15. It's about 15 centimeters. Okay? And then you're going to mark that onto your table for tube 1. So I'm going to write 15 centimeters. Okay? And then what's going to happen is we're just going to go through and do the same thing to each test tube. So here is tube 2. Yeah. Mixing up your yeast. Okay. Putting it into the test tube so that it goes to the top. Taking my glass test tube and pushing it to the very top. And then twisting. I spilled a little bit, and that's okay. Okay, putting it into my test tube rack. All right, and I'm recording my initial gas heights for all of them. And then here's 3. So once you put the yeast in with the sugar solutions, it should start to ferment. And what you will be seeing is the release of a gas when that fermentation occurs. Great, so there's number 4. Remember, measuring the initial height each time. The initial height should basically be the same, because you have, you know, the same amount, you should have the same amount of liquid in each test tube, because you're filling it all the way to the top. So don't be worried if the initial height is the same. All right, so here's our last one. Okay, so now we have all of our test tubes fermenting, and what you want to do is let the test tubes sit in a warm area. It says to put it by a window, but this is Colorado, and whenever you're doing this, it could be 50 degrees outside, it could be 30, it could be 80. So I would not put it by a window. I would honestly just put it in a warm part of your house, and let it sit there for 2 hours. I know some people have put it in their laundry room because they said that their laundry room is the warmest room. And then what you're going to do is you're going to come back, and for each one, you're just going to measure that gas height again and see how much it has changed, and how much fermentation has taken place. So that's it. After that, while you're waiting, the best thing you could do is start researching what are the ingredients in Equal and in Splenda and in regular sugar, and how does yeast and sugar interact with each other? Like, what does it do? What does warm water do to yeast? What does the yeast use the sugar for? Because yeast is alive. And so what is it doing with that sugar? Because that's going to become important whenever you're completing your analysis questions and in your conclusion. So while you're waiting for that to ferment, that's probably what I would do, is I would start doing some internet research about the different types, different ingredients in these different sugars, and how do they react with yeast, and what does the water do with the yeast. So I hope you enjoyed this and that it was informative and that it helps kind of alleviate any fears you have of doing the lab, and I will see you later. One last thing that I wanted to add: when you are done with everything, you want to rinse it with soap and water really good. The pipettes, you can, I would keep them, but I would take a glass of warm soapy water, and I would write like pipette up and down to get them clean on the inside. And then I would put them in a bag that says used pipette, so that way you know, hey, I've used these and there might be a little bit of residue on them. Your kit came with 20 pipettes, so you should have enough clean ones for all the different labs, but just in case, I would keep them. Keep your solutions, and keep your yeast. I would just put it in a bag and store it in a dark place, because you are going to use yeast in the other labs. But the test tubes, all the lab equipment, you will use that again. So don't get rid of it. Just clean it really well. Fermentation Lab, and I have let my test tubes sit in a warm area for 2 hours, And so what you should see is differences in the bottom of the liquid and the gas. So I'm going to bring one kind of up close to you. And so you can see that it's actually still reacting. There's bubbles coming up. And so where you want to measure is that bottom part of the liquid all the way to the tippy top of that small plastic test tube. So this one was about 17 millimeters whenever I measured it. So a couple of things. Some of the test tubes may not have hardly moved at all, whereas others are only going to move up from your initial height like 2 millimeters or like 1. Okay, so we're not going to see like drastic changes. You're just going to see little increments. And the other thing I would highly recommend is that you place it in a really warm area. If there is sunlight when you're doing this, put it in the sun, like inside, not outside. Don't put it out in direct sunlight. Or have kind of like a lamp near it, just so that way it'll actually heat up, because you do need that heat to start the reaction. And so then what you'll do is you'll just record, you'll take your ruler and you'll record each gas height as your final gas height, and then you'll note the net change, and then you'll see which one fermented, which solution fermented the yeast the best. And then you'll need to explain why.